Okay, let's get started, everyone. Thank you all for participating in the breakout sessions. I was walking around, I went through all the sessions, and really happy to see very engaging conversations in all of them. And now we get to the readout session, very exciting, so that we can now hear from what came out of each one of these sessions. We have until 3:45, so I would like to ask each breakout lead who is presenting the readout to please restrict your remarks, your report to about 8 minutes to allow a couple minutes for questions from the audience. So let's get started. We're going to start with the benchmarks breakout. So. All right, so I see Pat, you are doing the readout?
I think so.
Thank you very much. All right.
It'll be way less than 8 minutes. So we had a very lively discussion about benchmarks. It was great. I think one of the things that we agreed on is that there probably are data sets out there that could be used for benchmarking that just aren't. So what we need is some mechanism where we can start to collect these and let the community know where they are. So there are a couple of efforts around this. There is a more general effort with the AI Alliance, which is collecting data sets. We have another more drug discovery oriented project that I've started working on with a few colleagues from other pharmaceutical companies and biotech. So we have an effort called Polaris. So if you go to polarishub.io, we have started to collect data sets there and make them available to people. In addition, we're providing expert level approval of these data sets. So we're getting domain experts to look over the data sets. So this was another thing that we agreed on and in our breakout session is a lot of this data is very domain specific and we need to have experts who can curate the data and take a look at it. Next, we really need methods, standard methods for doing data curation. So we need workflows that will go through and capture a lot of the standard mistakes that occur in these data sets and can correct those or point out or point to them. And then finally, we need to get to the point where we have standards for evaluating and comparing the methods. So I feel like we came to a relatively solid conclusion on data sets where we're simply trying to make a prediction. You've got a chemical structure and a label, you're trying to build a model. That was relatively straightforward. Then we segued a little bit and started talking about generative models because there's this whole emerging field of molecule generators and neural networks and various other approaches to molecule generation. And how do we benchmark those? Because this becomes very complicated because you're trying to benchmark creativity. And really, I think the only solid conclusion that we came to was if you can do experimental validation to show that your model is generating something that meets some experimental objective, that's great. But that's also expensive and time-consuming. So I think the jury is still out, and we didn't really come up with great, here's a benchmark that you can use. There are lots of criteria that have been established in the literature, but a lot of that doesn't get you back to, is this generating an interesting molecule? So I think there we kind of threw our hands up and said, the field is still evolving and we'll have to see what happens. So that's benchmarking. Sure.
So thank you. That's a lot of great directions that was pointed out for us. Now, if if I were to ask, what do you think would be a good first project to get us started along these directions? What would you say? Did you come up with some recommendations there?
Yeah, yes, absolutely. I think there are a few things we can do that would really help to move us along. One is coming up with places that the community can go to deposit data sets, to share data sets, to make data sets available for expert evaluation, and then just putting together workflows that everyone can adopt, all the way from doing curation, building models, comparing models. So I think having standard data sets, having standard methods for curating those data sets, and then standard methods for comparing models that are agreed upon by the community.
Great, great, thank you. And I would like to thank everyone who participated in this session. Okay, next we have multimodal fusion.
Okay, so we took on the task of sorting out where we want to apply multimodal fusion. We talked about early, mid, and late fusion architectures and their advantages in particular data sets that Alex Shalek brought to the team, so he could describe that now.
So why don't you describe what our task is? I'll describe the biology and we'll go from there.
Okay, so the purpose of this task, it was a very large team of energized and excited students primarily. We think that the goal that we've converged on is to establish a kind of test bed for multimodal integration across multi-domain tasks. These can be tasks related, as Alex will tell us about, tumor, human tumor and cancers. These can be modalities spanning multi-omics to imaging, and they will extend these multimodal solutions from simple regression into sort of a network of models for solving more complex variational inference problems. The team thinks this is ambitious for 12 months. We set our milestones accordingly, but the statement remains. This is sort of the vision that we're thinking of pursuing in the space of cancer. There's a lot of reasons why this is needed. Alex is very familiar with this. It's the focus of his lab, so I'll let you speak to that. And ultimately, you'll see the milestones are technically aimed at sort of a generic baseline for some multimodal tasks to be determined and to produce open releases of the test bed and framework for those, first internally and then on Hugging Face. Alex?
Great, thanks, James. So as James highlighted, there have been a lot of studies as of late that have focused on generating single cell or imaging-based data and then applying computational models to them and doing pre-training there. We got really interested in whether some of the emerging data sets that have come out of the Human Tumor Atlas Network would give us an opportunity to apply these approaches and potentially to use multimodal fusion to bring together things that are usually not analyzed in conjunction. So say H&E images or things like single-cell genomics data. And so things that we thought could be great were to think about ways of creating a multimodal joint embedding and predicting values where they were not measured as a way of extending and enriching available data sets, which would be incredibly valuable to the cancer community.
Next slide, please.
I got it. Yeah, it's on my hands, James. It's a good thing you weren't at the microphone, otherwise it would sound really loud. So although we tried to be very ambitious in the initial conversation and very ambitious in the idea of creating 2 slides in a matter of an hour, we were discussing things in 15 different languages, we came down to what we thought was identified by the group. We thought a reasonable milestone for about 3 months was to identify 3 distinct modalities and open AI models that have been pre-trained on similar modalities. And there will be a few of these. We brought up multiple different options for things like single-cell RNA sequencing. All of those can be brought to the table. And in parallel, we'd like to go into the available H10 data sets to figure out which are the ones that are really useful for fine-tuning. What might it look like? An internal Teams page. I hate Teams. An internal page of something that would enable us to share information. I'm allowed to say that. And really be useful to the computational community, I would say, particularly for people that are thinking about masters or PhDs. And the real goal would be to figure out if there are ways of creating mid-fusion strategies using it. I think by 6 months, we'd like to create a prototype bed that would bring those together, where we would think about how that might all look, and talk about pros and cons of different architectures as envisioned by different individuals. And I'd say 12 months, we'd really like to test it, and I think that testing some of these different configurations and comparing them could lead to joint publications or proceedings. And I don't know, James, if there's something you wanted to add?
Yeah, sure. So in the previous slide, we showed our email addresses. I think they show up. Oh, they didn't, so we have to reload that. But please come up to us after, get our contact information. If you were in the breakout session or if this session seems interesting to you, we'll make a list. We'll start to aggregate that and schedule follow-up meetings. The last thing I wanted to point out is we don't think of this breakout session as independent of the other ones. So the last row, we think that there are architecture questions that the architecture breakout session will discuss that might inform how we go about building this multimodal testbed. We think of the challenge and competition breakout session as providing potentially a way for us to use the human tumor atlas network data towards a particular goal, publication, proceeding, or competition entry at the 12-month stage. So anything else, Alex?
I would just say thanks to the people that joined the breakout, and apologies if we did not capture what you intended, and if we did, we're glad we did.
Thank you.
Thank you.
Thank you. That's great. Okay, next. Thank you very much. That was wonderful. Next, we have the model architecture.
Okay, so as I mentioned before we start, we use the term architecture very broadly, right? So we discuss about how we can look at all those different data representation, different model architecture, and the training methodology, anything that can help us moving this field forward, right? So before we start, we did a really quick poll to see who our participants are, and 100% of them are all AI, ML, data scientists, and we don't have anyone who has a very strong biological background or more translational research. But this maybe some people who do have, but did not respond, but that's the group we have. And also we did a really quick poll to see how people are familiar with those very well-known models, right, like DNA bird and C-bird, something like those. And you can see that a lot of people are still more familiar with the protein world. And then we have a very young and energetic group. They are willing to do anything. So I kind of forced them to say, hey, we need to come up with some workable next steps, right? So how about we do single cell? And everybody agrees. So the idea of that is we talk about a lot of that today, right? So this is a recent paper talking about the AI virtual cell, and the virtual cell idea is not new, right? It has been there at least for 20 years. And then now we have a lot more data, we have a lot more computational power. We think this may be a good time to do it again. And then the idea is we have those different data modalities, and we train foundation models and to see if we can do a lot of those in silico tests much faster than before, right? And also at the same time, we see a lot of papers saying, hey, you know, your model is not, you train on so many data sets using so much resources, and your model is not really better than our very simple linear models, right? So especially in the gene perturbation effects, this is a very recent paper saying, well, I just do a linear head, and then it's the same or even better in when I compare it to gears, SCGPT, and SC transformer, all those very well-known ones. And so what's the problem, right? So as a group, we decided maybe we can do something to begin this journey, right? We want to do something in the open sourced foundation model for gene perturbation prediction. And then the idea is we want to open source, so actually at IBM, we will also open source our single cell model and DNA models. That's something you can use. And also feel free to use anything, right? You can use the CGPT or any model that you are developing. And the idea is there are so many different levels of data sources, of DNAs, of RNAs. How do you use all those together and to train a model, right? So release your model, we'll release ours, and then as a group, right? We talk about how those models should work together, and that's the initial goal. And then we can work with the benchmarks, right? So we want to look at the current perturbation data set. And then we know there are issues with that. They are very biased, and they are you only have that for very small data set, right? So we want to work together to find the good ones and what are the really good metrics when you compare the result. And then that is our 6 months goal. And at 12 months, we can work with the challenges and competition group, and then we want to host a challenge using the benchmarks and then the models we identified, and hope everybody will participate and try out your ways of modeling the data, your ways of pre-training, your ways of fine-tuning, and see if we can extract enough information from the pre-trained model to beat the simple ones.
We actually are related to everyone. Everybody who wants to do a challenge, benchmarks, architecture, models. Everybody welcome.
Yeah, so we had a great discussion. You know, at the outset, Pablo presented an overview of his experience constructing and running prospective blind challenges, primarily in the areas related to systems biology, and the principles of data blind prospective challenges is the best way to uncover weaknesses in model training and evaluation. And so a lot of our discussion focused on brainstorming the intersection between the types of data sets that are amenable that we could imagine gaining access to that could be embargoed and allow a challenge to be run by the community and then unblinded and the results revealed, and also intersecting with areas of high impact for essentially small molecule drug discovery. And so, as an example of some of the kind of initial points of discussions, a lot of our ideas tended to focus on questions of target engagement. So, there are data sets. Leash Biosciences recently published a challenge related to DEL screening data. An attendee, Liliana Guerrero, was describing a fragment X-ray screening data set. We think about one of the key, actually this must not have updated, made some minor changes, but one of the key questions that some of the co-folding models might be particularly amenable for is predicting the binding of compounds to cryptic allosteric pockets. So can you predict the conformational changes upon compound binding? And so you could imagine a challenge where participants could make predictions of the site of binding, make predictions of which fragments or compounds bind, and go even a level further and try to predict the functional impact or consequence of that binding. And the data sources related to that, the Center for Multiplex Assessment of Phenotype listed there, for example, was a repository of deep mutagenesis data. And then, additionally, in terms of molecular interaction, data sources from Dana-Farber, for instance, of molecular glues. And, ultimately, near the end of the discussion, we were thinking of analogous government-funded programs, such as the D3R challenges that used to be funded, I believe, by NIH. And so, I think our idea would be to, use this as an excellent starting point, define the potential challenges and specifically the key question, and use that as a starting point to, initiate a formal challenge that would be announced at ISMB Liverpool in 2025. And so, ideally, in addition to a well-defined data set and a key question, we'll also have some essentially baseline models, null models that can be recommended and used as a sort of standard across all of the community of participants. So I don't know if there's anything you'd like to add to that.
No, yeah, just that. I mean, we're really the big problem is as Pat also said, is find the data set. I think we started with it was a very good start because people already had suggestions, so that was great. Thanks for the participation. Also, one of the big focuses, of course, it's not just a normal challenge, right? We really want to push the idea of using open source foundational models for trying to solve this challenge. And so that will also probably be part of the organization, which would be, where do our existing models take us with solving these questions, and how can we go further? Are these really good data sets for benchmarking foundational models or LLMs? Because the types of problems that you find is that if these are trained with all the existing data, maybe there's some leakage already for trying to solve this question. And so DREAM is organizing at ISMB Liverpool a specific session on benchmarking foundational models. And so we really want to take that advantage, and I invite all the different also breakup sessions to if they come up with a challenge with a data set with a problem, we can include it in that session as that focus on foundational models or LLMs for trying to solve these problems. So thanks everybody for the participation.
Thank you.
Okay, thank you, that's fantastic. And thank you to all the breakout leads and everyone who participated in those. And it's really great to see what's reflected here is a lot of in-depth discussions went on, and very importantly, a lot of very concrete ideas in terms of projects where we can get started with came out of it, and which is really exactly what we were hoping for these breakout sessions. So as James pointed out, these ideas will be followed up. We are going to organize projects within the AI for Drug Discovery Working Group, within the AI Alliance. So there are whiteboards out there, as some of you have put your stickers there already. If you were participating in one, but now after hearing the readout session, you decide, oh, I want to participate in that project instead, feel free to go move your stickers, that's allowed. If you didn't put your sticker up anywhere yet and now you're convinced that you want to join one of the projects, please go ahead and do that as well. We would really like to see a lot of participation in these projects. Again, these are really great projects that came out of these discussions, and great starting points for us to go forward with the working group. So thank you again. So we have a break after this, and then after that, we have saved the best session last, which is the hands-on demo and lab, which starts at 4 p.m. But since we are ending this one relatively early, you are most welcome to get there early, after you grab some coffee and some refreshments, and maybe take this time to register if you haven't registered yet. But again, it's going to be really exciting, where you are going to experience firsthand, some of the models, latest AI foundation models that have been open sourced. And you'll be able to walk through the steps of downloading the models, building on the models, and walking through the use cases, and then in the end see a demo of how these models can be used in a real-world drug discovery workflow. So I would like to encourage all of you to participate. And with that, thank you very much, and see you there. And one more thing, after that, please do come back here again at 5. Where we are going to have just a couple of closing remarks, followed by a network reception. Yannis, do you have anything? Okay, great. All right, see you back there.
So if we can have your attention for a very short period of time, we are very much aware that we are behind, food, so we'll be very short. So I'll ask, thank you. So we have some very short, brief final comments. So first, I'll ask Oran Krieger, who is a professor at Boston University and also is the director of the Mass Open Cloud Alliance, and he's currently also affiliated with Red Hat, to say a few words about the Mass Open Cloud Alliance and the New England Research Cloud, because there were questions that came up during the hands-on session. So, Oran.
Hey, everybody. So I just wanted to kind of, how many people did the demo session just now? Raise your hands. All right, so the Mass Open Cloud has been this project that's been going on for a long time. Red Hat is recently decided with the focus on AI, they really want to much more deeply invest on this as a way of reaching out to academia and to science, where a lot of this AI is happening. And so I'm here now at Red Hat, working with Heidi, wherever she is, who does all the real work. She's somewhere back there, I hope. But to kind of lead Red Hat's efforts around this. Even though IBM owns Red Hat, we're really kind of two separate companies working together on this project, which was really cool. In this case, the idea behind the Mass Open Cloud was, let's build a not-for-profit cloud operated and run by universities. So currently it's Harvard and Boston University that are in charge of the operations on this. It's nonprofit. We charge research and education nonprofits, with exception, since it's hosted in the Commonwealth, also startups from the Commonwealth, the real costs of using the cloud. And what's so cool about this demonstration from IBM is this is a real science use case where the entire stack is deployed on this and available. So hopefully people will continue to use this, but also use the Mass Open Cloud themselves and set up an account after this, and reach out to us if you have any questions.
Okay, I have also very short closing remarks before Jianying talks. I would like to thank you for joining us today. There are many things that are due, particularly to my co-conspirators, Jianying, Diane, Feixiong, and Mikal, that are here. This was actually an idea of Jianying. We started working on this in the summer, and I thought it was a great event today. I would also like to thank the Boston University administration for supporting this, from the provost to a number of different offices, the industry engagement, government affairs, and also thanking the Mass Open Cloud Alliance and Red Hat, particularly Heidi Dempsey, Phil, Nancy Clinton, Tara Moran, who did a lot of work behind the scenes, particularly in setting up the hands-on session today. And also the facilities people that made this possible, and the AV crew. And finally, and last but definitely not least, I would like to thank Catherine D'Angelo, that did a great work organizing this, and the Hariri administration team that together with her pulled this event together. So with that, I'll pass on to Jianying for closing remarks.
Thank you, Yannis. I would just first really second that call out to Catherine and her team, right? I have been to many, many meetings. I think I speak for a lot of people here. This is one of the most smoothly run meetings that I have been to. So thank you so very much. I know there's a lot of work that went behind it. So again, thank you. And then I would just like to close by thanking all of you who came here, and really, I have been really touched by the level of energy, the level of engagement, and what came out of it. And so thank you very much. Thank you for the active participation, and thank you for your enthusiasm, and thank you for your energy. And I'm coming away from this really, really excited and energized, and looking forward to the follow-on work, and really ready to go. So I hope you all feel the same way, and enjoy the networking session, and see you.